So good morning, everybody. My name is Matt Edick. I'm the director of the Center for Strategic Health Partnerships at the Michigan Public Health Institute, and I'm also the director of the Midwest Genetics Network. I'd like to take a moment this morning or afternoon to welcome you to the Virtual Learning Collaborative focused on improving care for developmental disabilities and dysmorphic features. It's been brought to you by the Midwest Genetics Network here at MPHI, in partnership with the Minnesota chapter of the American Academy of Pediatrics and the Minnesota Medical Association. Excuse me. The Midwest Genetics Network is a 7-state network funded by the Health Resources and Services Administration and focused on improving access to genetic services for medically underserved individuals. This continuing education and quality improvement project is one of many resources developed by the Midwest Genetics Network's stakeholder education and engagement work group. So today's presentation is going to be on an introduction to commonly recommended genetic tests, and it is the final didactic webinar in this 4-part series that's been going on for about the last 4 months. And for any of you that are participating in these webinars to earn MOC4 credits, just for your information, there'll be one more follow-up webinar, kind of a debrief and an opportunity for you to share some of your learnings. But in terms of the didactic presentations, this is the last one in the series. So just a couple housekeeping things before we get going. We will be sending out a post-series assessment today after the presentation. It's really important that we have you all complete that for us. There's 2 reasons for that. First of all, the data that we collect in the pre and post assessments is the data that we use to justify to our funder to get more funds to do projects like this and offer them to you for free. So we really need that data and really appreciate you taking the time to provide it for us. Also, for any of you seeking continuing ed credits, so CME or MOC2 credits, we can't issue you your certificate until you complete that assessment. And so once you do complete that assessment, we'll be able to send out your certificate. So with that, it's my great pleasure to introduce our speakers, our speaker, Ms. Sarah Zorin, and our moderator, Dr. Jess Scott-Schwer. Ms. Zorin is a certified genetic counselor at the University of Wisconsin Weizmann Center Medical Genetics Clinic and has been the coordinator of the Genetic System Integration Hub since 2016. Ms. Zorin earned a bachelor's degree in genetics and a master's degree in endocrinology and reproductive physiology from the University of Wisconsin-Madison. She's worked in basic research in academia and is also a longtime member of the Midwest Genetics Network Stakeholder Education and Engagement work group that brought you this series. Dr. Scott-Schwer is an associate professor at the University of Wisconsin Department of Pediatrics, Division of Genetics and Metabolism. She completed medical school, pediatric residency, medical genetics residency, and a biochemical genetics fellowship at University of Wisconsin School of Medicine and Public Health. Dr. Scott-Schwer is a key partner in several Midwest Genetics Network initiatives and also was recently appointed to the advisory board for the Midwest Genetics Network. So without further ado, I would like to turn this over to Ms. Zorin to tell us about commonly recommended genetic tests.
Thanks, Matt. I can't believe I left this off my bio, but I did obtain a second master's in genetic counseling, so I am a certified genetic counselor as well. So if we could actually have the next slide, please, Danielle, thank you. So Jess and I do not have any conflicts of interest to disclose. The next slide then itemizes our learning objectives, which that the participants will be able to describe the basic differences between commonly recommended genetic tests, as well as describe possible test outcomes for commonly recommended genetic tests, and then to describe the value of pre-testing genetic counseling and informed consent. The next slide, please. Oh, there we go, thank you. So as Matt said, this webinar is really intended to be a high-level overview of commonly recommended diagnostic genetic testing for individuals who are affected with various neurodevelopmental disorders, specifically developmental delay, intellectual delay, and autism spectrum disorder. And specifically, we're looking at the identification of germline variants for Mendelian disorders. And Mendelian disorders are defined as inherited disorders that are caused by a pathogenic variant in a single gene. And this is as opposed to other disorders which may have a more complex genetic signature with or without several or one or more environmental influences. And so the tests that you see on the screen are the ones that we're going to be talking about today. I have grayed out the genome sequencing. We'll talk about it a very little bit, but it is not commonly recommended at this point. And so just so that you know, we'll touch on it a little bit. The next slide, please. I wanted to, with the first two slides, provide some foundational information that will help hopefully with the rest of the presentation. And the first slide really is to talk about some definitions. So the definitions for the terms variant and polymorphism and mutation are sometimes blurred by some. And so you can pause it right there for a minute, Danielle. And so what I wanted to do is just to talk a little bit more about them and hopefully pull out some of the distinctions. So a variant is simply defined as a change in the genetic material that's compared to a reference genome. There's no implication in this definition of whether this is a harmful or benign change. And when we think about variants, they can really occur on very different scales, so anywhere from an entire chromosome that could be either extra or missing to a portion of a chromosome that may include hundreds of genes to just a handful of genes. We can have variants that include parts of a gene or several nucleotides or even just down to a single nucleotide. So there are many different scales of variants, and for the most part in this presentation we're going to be talking about two different classes of variants. So either nucleotide sequence changes, we would call that a sequence change, or where there is extra or missing pieces of genetic material of various sizes. So this we sometimes refer to as gains and loss, deletions or duplications, and also sometimes we refer to them as copy number variants. Next slide, please. So the term polymorphism then it's actually defined as a type of variant. So it is a change in the genetic material, but it is a type that does occur in greater than or equal to 1% of the population. And typically the consequence to the phenotype is either neutral or beneficial. And so we do use modifiers such as benign or likely benign to describe polymorphisms. And a mutation, on the other hand, occurs more rarely, so less than 1% of individuals in the population will have that specific change, but the consequence to the phenotype is typically harmful. And so there are modifiers that we will use, such as pathogenic or likely pathogenic, to describe these kinds of variants. I think it goes without saying that we all have variants, probably 100,000 variants, which is what makes us unique from each other. Next slide. And to know that variants can be inherited from a parent, or they can occur for the first time in an individual spontaneously. And so we would refer to that as either being acquired or being a de novo variant. Parental testing is required to determine whether a variant is inherited or de novo, and we will use this information then to establish recurrence risk estimates for the individual or the family. Next slide. For the vast majority of the tests we're going to talk about today, a bioinformatics pipeline is used for variant identification, analysis, and classification. A downstream element in the bioinformatics pipeline is variant analysis. And this is where queries are made into multiple databases to extract information about the variant and then prioritize candidate causal variants. To give a little bit more information about what I mean by that, so for each variant that is identified, these databases will be queried to identify information such as, has this particular variant ever been reported in another individual? What is the frequency of the occurrence of this particular variant? If the variant has been reported in other individuals, are they healthy or do they have some kind of medical concern or features? If they do have medical features, what specifically are they? There are also databases that we can query that will, or the test labs can query, that will look at computer models that will predict the effect of the variant on either protein function or cell function. And there's also databases that will give us information on inheritance and a whole slew of information. But very importantly is another line of inquiry, which is using the phenotypic information that was submitted to the test lab by the ordering provider. And this is submitted in what we call HPO terms, or human phenotype ontology terms. And the HPO terms are essentially a standardized vocabulary that is used to describe phenotypic abnormalities. And one point that we have made throughout the presentation, which I'll make now and then at least once more in this presentation, is the importance of obtaining a complete and accurate phenotype of the individual being tested and then submitting that information to the test lab. And that is because this information is used in the variant analysis process, and so this process is phenotype driven. The variants are then classified based on the strength of available evidence along a spectrum of pathogenicity using a quantitative evidence-based scoring framework. Next slide, please. So on one end of the spectrum, variants can be classified as either benign or likely benign, and this is where there is a lot of strong evidence that is suggesting this particular variant is not causing any medical concerns in the individual, and so the test lab will report this as either normal or negative. On the other end of the spectrum are variants that are likely pathogenic or pathogenic, and these are variants where there's a lot of strong evidence that this particular variant is causing medical concerns for the individual, and it's really matching up with what we are seeing in the individual. So that would be an abnormal or positive result. But in the middle of the spectrum is this this variant classification called variant of uncertain significance. We sometimes refer to this as a VUS or a VOUS. But this is a situation where there's either not any evidence or not much or not enough strong evidence where we can, with any degree of certainty, classify this variant as being either benign or pathogenic. And so we have to leave it in this very uncertain spot. And this particular result can cause there's a lot of misunderstanding with not only families but also other providers about what the significance is of a variant of uncertain significance, and whether diagnoses are made from it and whether medical management should or shouldn't change from it. And so we spend a lot of time counseling on the likelihood of finding variants of uncertain significance. And it's also important to know that the classification of a variant can change over time as new evidence becomes available. And so with variants of uncertain significance, we do believe that over time they will be reclassified as either benign or pathogenic. But it's also important to know that a likely pathogenic can later be downgraded to a likely benign variant, or the other way around. And this isn't because there was a mistake or error made in the testing process or in the pipeline. It's simply because we've learned more about that particular gene or that particular variant over time. Next slide. So with that in mind, I wanted to dive in, and starting with the microarray-based genome copy number analysis. So this is a technology for first-tier genetic test for individuals with DD-ID and autism spectrum disorder, as well as individuals with multiple congenital anomalies that are not specific to a well-delineated syndrome. And in a real basic sense, it's a method for detecting of genomic imbalances. So the entire genome, all of the genetic material is assessed, and determining whether there are pieces that are extra or missing. Next slide, please. There are two related technologies that you may have heard about. One is called comparative genomic hybridization microarray, or CGH microarray, and then also, excuse me, the single nucleotide polymorphism, or SNP arrays. An important distinction between the two is that the SNP arrays are able to detect something that we call uniparental disomy. And this is a situation where an individual will inherit both copies of a chromosome pair from the same parent. So normally what happens is that an individual will inherit one chromosome from one parent, and the other chromosome of the same pair from the other parent. But in some cases, the individual will actually inherit both chromosomes of a pair from the same parent. And that's known as uniparental disomy. And this is involved in certain imprinting disorders. And you may be familiar with Prader-Willi syndrome and Angelman syndrome are two that are involved with this kind of inheritance. The next slide. Additionally, with SNP arrays, there are some unanticipated findings. So importantly, consanguinity, which is when the parents share a common ancestor, as well as non-paternity. Next slide. And so with this testing, it really is important to inform the family or the individual about these possible results before testing and before the sample is obtained. The next slide. So with the microarray technology, similar to what we were talking about in the previous slide, the variants are identified and analyzed and classified in this pipeline along a spectrum of pathogenicity. So the next slide then is just showing an example of a finding that we may get. So this is an ideogram of chromosome 22, which is one of our smaller chromosomes. The top little blob is representing the P arm, or the petite arm, the small arm of the chromosome, and then the Q arm is down below, that's the longer arm. And in the box is the result, the abbreviated result that you might find on a microarray report, where it's showing this is an abnormal result, and that on the 22nd chromosome, on the Q arm, or the long arm, at the position 11.2, there was a loss of material that was 3 megabases in size. And after the variant analysis process, it was identified that this is a recurring microdeletion syndrome. It's well known what the genes are in this region, as well as the phenotypic features, that they're very well characterized, and that in fact the features that have been reported in the literature line up very well with the features that were submitted to the test lab of the individual being tested. And so this particular variant was given the classification of pathogenic, and this is consistent with the diagnosis of DeGeorge syndrome, which is also known as 22q deletion syndrome. Next slide. The Fragile X by PCR with methylation studies is another first-tier test for individuals with DD-ID and autism spectrum disorder. And it is used to diagnose or rule out FMR1-related disorders. FMR1 is the name of the gene. It is located on the X chromosome, and the three disorders in the FMR1-related disorders are Fragile X syndrome, which is the most common cause of inherited ID and monogenic autism in males, but also Fragile X-associated tremor and ataxia syndrome and Fragile X-associated primary ovarian insufficiency. Fragile X and the FMR1 related disorders are a different kind of variant. So we previously were talking about nucleotide and deletions and duplications. This is in a class of variant known as a nucleotide repeat expansion disorder. And so what are nucleotide repeats and how do they expand and how does that lead to disorders? So on the next slide, this is an example of the green box is representing a gene, and the nucleotide repeat is actually these single nucleotides of CGG that this sequence is repeated in tandem. So this normally happens throughout the genome, where there are places where we find these nucleotide repeats. But during gametogenesis, and specifically in meiosis, these repeats are unstable. And so in the next slide, you will see an example of how these repeats can then expand or grow. And this can disrupt the transcription of the gene that it is near or embedded within, therefore causing disease. And so these nucleotide repeat expansion disorders, there are probably about 40 of them. They primarily affect the nervous system. And other examples, in addition to Fragile X or FMR related disorders, are Huntington disease, myotonic dystrophies, and different ataxias such as Friedreich's ataxia. So in the case specifically of FMR1 related disorders, in the next slide, you'll see that this is actually representing the FMR1 gene, and the CGG is the trinucleotide repeat. And in the next slide, you'll see that when this expansion is big enough or long enough, then the DNA will become methylated and the gene is inactivated, leading to disease. And so in the next slide then, what we have is that this test, the Fragile X by PCR with methylation studies, actually determines the number of the CGG repeats and the methylation status. And this has been very well characterized and correlated with risk for developing disease as well as the diagnosis of Fragile X syndrome. The next slide is just intended to be a reference slide. I'm not going to go over it in any detail, and it is available for you to download as a PDF, but it's just trying to summarize the number of repeats as well as the methylation status and how that correlates to risk based on the sex of the individual. The next slide. Moving on into single gene and multi-gene, which we call panels analysis. Just a real basic primer before we start that genes contain both coding regions, which we call introns, and non-coding regions, which we call, sorry, did I say that wrong? Coding regions known as exons and non-coding regions known as introns. And that for both single and multiple gene analysis, there is an interrogation primarily of the exons, but also some flanking intronic regions, and specifically looking for two kinds of variants, either sequence variants or deletions and duplications. And for a single gene analysis, this situation you may be considered when there's really a quite narrow differential. So an example would be analyzing the P10 gene, which is associated with several disorders, but they do have as a feature autism disorder. And I really, with this particular slide, the information that I'm going to talk about is really not intended to be like a prescription for when to order small panels or when to order large panels. More, I'm just trying to give an example because, truly the type of test that's ordered is really based on clinical findings and clinical judgment. And sometimes there are some other considerations such as insurance coverage and family preferences. So the next slide then is just a little bit about gene panels. And so with a panel, we're analyzing multiple genes all simultaneously. Panels vary in size. They can be anywhere from 2 genes to thousands of genes. But they typically all have something in common. So they could either, all of the genes on the panel could be associated with the same condition. So for example, ectodermal dysplasia, or perhaps a broad group of conditions such as neuromuscular disorders, or all the genes on the panel could all have in common a particular phenotypic feature such as seizures or ataxia. The gene content on any given panel will and can and does change over time, and the diagnostic yields for the different panels are also vary widely. And so sometimes panels are considered when there is a complex disease presentation, or there might be some overlapping clinical symptoms. Next slide, please. And as before, all of the variants that are identified are then analyzed and classified along a spectrum of pathogenicity. And it's very important to note that we can receive a positive, a negative, or a variant of uncertain significance result for each gene that's analyzed. And it's not uncommon if there's a larger panel that's ordered that we could actually receive a result where there are 4 VUSs and no positive findings. And that really illustrates the point that as an increasing amount of genetic material is analyzed, the likelihood of receiving uncertain results of uncertain significance will also increase. And so although with a larger panel you're casting a wider net, and there may be you may think that there would be a greater chance of obtaining a positive result, there was also the chance of obtaining uncertain results that may actually cause harm to the family by way of inducing anxiety or uncertainty or misunderstanding. So those decisions are very, very carefully considered. Next slide. Moving on to exome and genome sequencing. So again, just a few definitions. The exome is defined as the entirety of the exons. And in humans, we have roughly 22,000 genes, which equates to approximately 180,000 exons. And this is compared to the genome, which is defined as the entirety of the exons and introns. And just so for reference, the exome comprises about only 1% of the entire genome. So big, big difference in the amount of genetic material that's being analyzed in these two tests. But in both tests, we are looking at nucleotide variants as well as CNVs, although the technology is changing, and there may be, in some cases, ability to identify other types of variants, but I think that's a bit of a moving target right now. The samples that are analyzed are typically blood, but we can also obtain saliva and buccal samples as well. And I will say that, initially when we were ordering exomes, I think there was a higher likelihood or a higher instance of us ordering what we call proband only, meaning that just the DNA from the individual being tested was collected, and if there was a causal finding or something that was suspicious, we would then go and test parents to determine whether that variant was inherited or de novo. But I think now there is an increasing use of these duo and trio exomes, and that is where we're collecting not only our proband DNA sample, but also from samples from 1 to 2 first-degree relatives, most commonly the biological parents. And that is because there's a growing body of evidence suggesting that trio analysis will increase the diagnostic yield. So as before, the variants, the bioinformatics pipeline is used for variant analysis and classification, and then all of the variants are classified along a spectrum of pathogenicity. Next slide. With exome and genome, we can not only get our primary findings, but there's also a possibility of obtaining secondary and incidental findings. So the primary findings are defined as findings that are related to or possibly related to the reason for ordering the test. And these would typically on the reports, they are variants that are classified as either pathogenic, likely pathogenic, or VUS. We can also get secondary or incidental findings. And these are findings that are unrelated to the reason for ordering the test, but they may be of value. And so for these findings, parents and families are sometimes given a choice. And some labs have opt-in options and some labs have opt-out options. And so it's really important to read the fine print on the requisition and the consent forms. But the secondary findings are where the test lab will actively search and report pathogenic or likely pathogenic variants in any of 59 highly penetrant and medically actionable genes that are associated with various inherited disorders. So loosely, we call these the ACMG 59 genes. And some examples that you may be familiar with are the BRCA1 and 2 genes associated with hereditary breast and ovarian cancers syndromes, as well as the genes associated with Lynch syndrome and genes associated with sudden death due to cardiac arrhythmias, just to name a few. And so parents and individuals do have the option to get results from these secondary findings. Also, though, some labs will offer reporting of what is known as incidental findings. And so these are disease-causing variants, so pathogenic or likely pathogenic variants that will result in mostly adult onset Mendelian disorders, but sometimes childhood onset disorders. But the treatment or prevention may not be effective or available. And so again, very important to know what the test lab offers and to have thoughtful consideration about whether to offer this to the family or the patients. Next slide. Just to restate something that I said earlier, and that is that the likelihood of receiving uncertain or ambiguous results increases as the amount of genetic material analyzed increases. So the next slide is really the basis for the American College of Medical Genetics and Genomics putting forth a policy statement recommending obtaining informed consent prior to obtaining sample. Next slide. This is another slide that is meant to be a reference for you and which you can download as a PDF, and it's just comparing the different tests that we've talked about as well as their diagnostic yields. And this is, I should point out, is more specifically for individuals with neurodevelopmental disorders. The next slide, please. And to emphasize one more time the importance of obtaining a accurate and thorough phenotype and how essential it is for the genetic variant analysis, classification, and interpretation, and that the results interpretation is phenotype driven. Next slide, please. This is a paper that I mentioned in the second webinar, and this was a paper that compared the diagnostic yields between exome and microarray for individuals with neurodevelopmental disorders and found that the exome was outperforming the microarray. Next slide. And so we hope that soon the exome will be recommended as a first tier test, but that is yet to happen. And then the next slide is just to say that genome sequencing, although it is clinically available now, it is not widely used, and insurance companies, I would say probably with 100% certainty right now, are not covering genome sequencing. However, it is used on a research basis, and many undiagnosed disease programs are using genome sequencing. Next slide, please. I want to take a little bit of time to talk about the medical and case management that happens or could happen after genetic testing, and break it apart into so that it's on what kind of result we get. So if the result is positive, you know, I think that's our best case scenario. That's what we in genetics, what we hope we can achieve, is that we can provide a diagnosis and then provide a lot of information to the family. And so when we do have a positive result, we can provide that diagnosis. But even in that situation, sometimes we are not able to provide information on natural history or the clinical course or prognosis. And sometimes that's because we are still in our infancy in discovering a certain medical disorder or disorders and their associated genes. And so sometimes we can provide that and sometimes we cannot. However, when we do have a positive result, we are able to offer family testing to at-risk family members, and then we usually are able to provide some specific recurrence risk information. On the right end of the table, though, is when we get a negative result. And so although the result is negative, for some this can be actually reassuring because we may have been able to rule out certain conditions. But it's important to understand that a negative result doesn't mean that a genetic cause doesn't exist. And that is, you know, where we have to think about what kind of variants are we able to detect with the technology being used, and it is possible that the technology, well, we know the technology, one single technology will not detect all kinds of variants. So in the situation of a negative result, we may not be able to provide any information on natural history, but we still may be able to provide some information on treatment or medical management, especially if there is some family history involved. I think it goes without saying we wouldn't offer any family testing to other individuals. Thanks, Danielle. But sometimes we still are able to provide recurrence risk when we have a positive family history. And then with the uncertain results, you know, this is, again, I think, I can't say it enough how difficult these results are and why we spend so much time counseling on them, is because with that uncertain finding or the variant of uncertain significance, we aren't able to rule in or rule out a diagnosis. And so similar to the negative result, we may not be able to provide any information on natural history or clinical course, but sometimes we are able to provide information on treatment or medical management, and sometimes that's based on the personal and or family history. There are some occasions where we will offer familial testing for a variant of uncertain significance, but that doesn't happen all the time. And sometimes we are also able to provide some kind of recurrence risk information based, especially when we have a family history. And so the next slide is just another statement about what is not uncommon for us to see in clinic, is that when a VUS is identified in someone who has an undiagnosed clinical condition or clinical features, it can often lead to misinterpretation, misunderstanding, and this is not just with the family, it also happens with other providers, and also sometimes then there's inappropriate treatment, and certainly we can have unnecessary worry, and so we do spend a lot of time counseling on these uncertain variants. Yes, and variants can change over time. So with the next two slides, then, I just really want to take a couple steps back. I presented this slide in the second webinar, and that is, and I just want to touch on it again. We know there's an access issues in genetics, and so there may be some PCPs that are considering genetic testing, but we do recommend that that really be contingent on the ordering provider's ability to provide accurate and thorough phenotype information, as well as providing informed consent and being able to interpret and report the results. And so given the complexity of genetic testing, it may just be worthwhile to consult with a geneticist in advance to plan the testing and then interpret the results. And the final slide is really taking several steps back and just reflecting on all of this. So there is a historical connection between eugenics and medical genetics. And there have been, over time, populations for which we have lost trust, we have broken trust. And it's not a surprise to know that some of those populations to this day are disenfranchised with the medical community. And some are from underrepresented or vulnerable populations, and some to this day experience health disparities. And so we need to be very thoughtful when we are providing genetic services and ordering genetic tests, and we have to do better, and we have to rebuild trust. But that's a very long uphill journey. And there are ethical principles that have been established for responsible genetic testing and genetic services, but they aren't as easy to follow. So I think most people can agree that the risk of harm from simply drawing a blood sample or collecting cheek cells is quite low. But there is great risk of harm from genetic testing when you start to consider issues such as equity or emotional harm, other ethical considerations, financial harm, privacy issues. And so with the last slide is just a statement and a thought that I'd like to leave everyone with, and that is that when ordering genetic testing, equity and patient and family autonomy, privacy, emotional and financial implications, among others, should be considered and protected. So that's the end of the presentation. If anyone has any questions, we can open it up to that.
Thank you, Sarah. So I'm going to just turn it over to Dr. Scott Schwer to take any incoming questions. I see there's at least one in the chat there.
Yes, thank you. So what I see in the chat is the question, is it safe to assume that secondary findings would only be available for the patient, not parents, correct, if duo or trio?
I can answer, and then Jess, you can answer
Yeah.
too. It's not safe to assume. And that's one of the things that we talk about in the consenting process. So it depends on what the parents elect, that's one answer, but it also depends on the findings. So if a pathogenic variant is identified in one of the ACMG 59s, then the test lab will test the parents, and if one of the parents also has that variant, it will be reported. However, if one of the ACMG 59 variants is not identified in the proband, then nothing is reported about the parents. And so there can be the misunderstanding that if it's not reported in the proband, then neither of the parents have a pathogenic finding either. And that would not be a correct assumption.
And I think what that goes along with in talking about counseling and consent is you can see where this testing, both the primary finding we're looking for and then potentially secondary findings or incidental findings can really have family implications for parents, siblings, and then even, family members kind of outside that group, if you, talking about their healthcare in the future. I don't see any other questions in the chat. Any other questions that people have? Either put in their chat or unmute yourself and ask the questions. I agree. Sarah gave a very thorough talk and a lot of information to digest.
Yeah, I was going to say, Sarah, I think you might have hushed the crowd on this one. There's a lot of really wonderful information, and also just to be mindful of everybody's time. It is actually right at 1 o'clock, so we're right square on time. So there, I do see that one additional question just came in. Sarah, if you would, or Jess, if either of you would like to answer that, we can go ahead and do that. But then after that, we should probably wrap this up and let people go.
Okay, I'll read the question, Sarah, and then you could say something, and then I could say something if needed. So it says, how are you dealing with insurance issues regarding these tests? And that's actually probably another talk entirely. So.
Yeah, so I'm just trying to think where to start, because it is a big issue. So I would say for the first tier tests of microarray and Fragile X, I would say that most insurance companies will cover those for us. However, we do still obtain prior authorization. Beyond that, it's a bit of a crapshoot. There are some payers that we know will and some won't, but it takes a lot of time to write letters of medical justification, and even when it seems so very justified, we don't always get coverage. So it's I think a sore spot often for us. I think that it is getting better slowly. But there is definitely a lag in what the insurance companies understand about genetic testing and the usefulness of it, and then what they are able and willing to cover. Jess, do you have anything else to add about that?
I think you covered a lot of it, and I will say it is something that clearly in the genetics community, we know it is a barrier for our patients getting the care we feel they should get. And so we are doing advocacy around that, talking with payers and working to get improved coverage. So it's something we're trying our best to address, but it's a significant issue. And Matt, if you're talking, you're muted.
Yep, you are. It's just going to chime in.
See what I get for trying to be respectfully quiet? I was really just going to say we should probably go ahead and wrap up. We're about 3 minutes over, and definitely want to be respectful of everybody's time. So looking really quickly at the names, I noticed many of you on here have been on for all 4 of our webinars, and really glad to see that. But really just appreciate all of you participating, and thank you to our speakers and all the work that they've done in the team. And I know you can be on the lookout for your follow-up survey. And if you have questions or want to reach out, please don't hesitate. If you want to stay connected with MGN, please feel free to subscribe to our newsletter. I stuck that in the chat for everybody. And with that, have a really nice afternoon and nice day, nice week. And hopefully we will connect with you all again sometime soon.